Hello, this is our genetics section of microbiology. And what is genetics? Genetics is just a study of heredity from organisms. And our textbook gives us four different categories on this. And in a way, we need to look at them as like the first couple of them here, the transmission and expression of traits that are passed on from parent to offspring. And this is like the whole organism. And then the structure and the function of it, this comes down to the characteristics of the genetic information and its the biochemistry of the function of that information. And then four here would be how the genetic information changes through over time. Basically what we call mutation, which is inevitably evolution. All right, so genome for, depending upon the type of cell you're talking about, it'll be all the chromosomes. For a eukaryotic cell, it'll be the chromosomes and the mitochondria and chloroplasts. And then plasmids can be for a eukaryote. On a prokaryote, they're not going to have the mitochondria or the chloroplasts, but they'll have chromosomes and the plasmids. For cells, all living cells or all cells have DNA. We're talking about DNA. And on viruses, if you remember back when we talked about the virus in the virus section, it can be DNA or the RNA. All right. What is DNA? DNA is again the genetic material we see in the nucleus on the eukaryotes. It's made up of the nucleotides and proteins that these are that get the DNA gets wrapped around. In eukaryotes, these are histones, and prokaryotes, it's a different protein, and it also depends upon what species it is we're talking about. So it's a little bit different on prokaryotes than it is on eukaryotes. Another key thing is that in prokaryotes, almost all of them are a single loop or circular chromosomes. You see it right over here. And then they have the plasmids around them. That's DNA also. On eukaryotes, it's always linear like these here. Then they're going to have either mitochondria and or chloroplasts, and both of those have their own DNA. If you remember back from the eukaryotic section, we talked about the evolution of cells, and some eukaryotes also have plasmids in there. So there's lots of different DNA parts, or excuse me, there's different types of DNA in eukaryotic cells. And then in viruses, it's either DNA or RNA. All right. So what are we talking about when we're talking about genes? Genes are basically the part that's going to carry any the traits that are going to be transmitted from parent to offspring. And it's specifically a specific location on a chromosome. A chromosome has many genes on them. And we're just looking at a specific portion of that chromosome, which is going to code for a protein. Now there's three types that they can code for. We have the main ones that we code for proteins. This is, if you've ever done translation or transcription, which we will do in this is what we're talking about. Then there are RNA parts. So there's like rRNA, tRNA. There's also an RNA primer, and that's the genes for this. So it's a little bit different than the translation transcription, but we're mentioning it. And then we have regulatory genes that help, again, just like the name says, regulate our structural genes. And we'll talk about those in this unit also, this chapter also. So the genotype is whatever genes you have. The stereotypical one is be like the big B and the little b for a brown-eyed person, or and then they could also be the big B and big B. So both of those would be brown eyes, and to get little b, you would have the blue eyes. You'd have the two little b's. So these are one, and then this is one. But there's three different genotypes in there, because you have a dominant and recessive, two dominants or two recessives in this particular trait we're talking about. Then the phenotype is the actual expression, whether they have the brown eyes or the blue eyes. Typical Mendelian genetics you learned about in high school, most likely. So now about the gene sizes. All right, so viruses, some of this, I think there are some that might actually have two genes in them, but our textbook talks about them being 4 or 5 for the smallest ones. E. coli, they're showing you has basically 4,300 genes, and all this squiggly line that you're seeing out here, all of this stuff out here, that is the chromosome. That is the individual, the single circular chromosome of a particular E. coli. And if you were to stretch it out, it's 1,000 times longer than it is wide. All right. Or it's about a millimeter. No, that's not right. Anyways, it's really long compared to how long the size of the cell. And they give you the humans. Humans have 31,000 genes, and if you were to hold since humans are eukaryotes and it's linear, and if you were to hold one end of it and let it hang down, it would be approximately 6 feet long. So that's in each and every one of your cells. So it's DNA is pretty big, pretty long, I should say. It doesn't take up a lot of room when it gets all coiled and balled up around the proteins, but it's kind of long. What's DNA made of? DNA is made up of nucleotides. It's a polynomial, and which means it's made up of many parts, and the individual parts are called nucleotides. Each nucleotide on DNA contains a 5-carbon sugar and a deoxyribose, which means it's a ribose sugar missing one oxygen molecule. They have a phosphate in them. And then they have a nitrogenous base. Now, the nitrogenous base can be any one of these 4 bases. And that's going to dictate some of the information. This is where the information is. Information is found in these nitrogenous bases. All right. And if we were to draw one of these, let's say you would have a phosphate, comes down, you have a 5-carbon sugar, and there's a piece sticking off right here, and that's the 5' end. This right here is the 3'. That's because you number them. This is 1', because that's the 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. And they're telling you right down here that phosphates bond to the 5 carbon and the 3 carbon. So then there would be another phosphate here going to another sugar. So here's the 5, there's the 3. So it would bond this way, and it bonds to this 5 carbon here. And then off of this 1 is the base. And depending upon what type of base it is, it depends on the shape. I'm going to put thymine in there since we're talking about DNA. But this is the basic structure of a nucleotide right there, and what they look like. All right, so again here, the nitrogenous base goes to the 1'. And on adenine and thymine, this is very important. Adenine and thymine, there's 2 hydrogen bonds that hold them together, and they're showing you right over here. Adenine and thymine, there's 2 bonds holding them together. So there's that 5' I was talking about earlier, and there's that 3'. There's the 1', and you'll see it 1', 3', 5'. And down here, the guanine and cytosine, these have 3 bonds. So look right here, guanine and cytosine, there's 3 hydrogen bonds. Because of these bonds, it'll help twist the DNA, but these 2 bonds on the A's and T's will come into play a little bit later on in our discussion. Now, these run anti-parallel to each other, which means they're not identical. They're anti, which means they're opposite, and they run arrows. So for every A, you have a T. For every T, you have an A. So they're opposites, but they're different. And that's what that anti-parallel is. And when they talk about the 3 to 5, we're showing you here the 5' sugar. So that's the 5' carbon on the sugar. That's the 5' end. So the opposite side is that 3' end. On the other strand, they are in the opposite direction. It's the 5' at this end, and there's the 3' up at that end. So that's the anti. They're going opposite directions with each other, but they bond in a specific way. So when we talk about anti-parallel, that's what we're mentioning. All right. DNA is really just a template for information. And because of this, we can make exact copies of new strands based on the sequence of DNA, because A's bond to T, C's bond to G, so forth. So that enables us to make an exact copy of it. The significance of this is that because they do bond in certain pairs, it guarantees that the code will be retained. There's no changes. It can only happen in certain orders, unless you have mutations, but we'll get to that later. Now, the variety in this is that these genes can be in various orders of A's, T's, C's, and G's, and they can be various links, and that gives us a big variety. So that's the importance of the structure of DNA in that aspect. So our first concept in DNA, adenine is the complementary base for blank, and cytosine is complement for blank. And so basically we're talking about what bonds to adenine, and that is the thymine, and which one bonds to cytosine, and that is guanine. So in the process of replication for DNA, the original copy unzips, and that's this blue one, okay? Based on these pairs, again, for every G, we're going to have a C. For every A, we're going to have a T. So that's how we can make our exact copies. In the new copies, though, in the new, the blue one here is going to be our template to make our new our complementary strand. And this will split up into 2 different chromosomes. And on each chromosome, so here's the new gene or chromosome, the DNA strand, and there's the new one here. Half of it, the blue one, is the original, and the pink or magenta is the brand new piece. And in doing that, that's what we call the semi-conservativeness. Half of it will remain as the original, and you build the new strand. And that's what the semi-conservative process is of making these. And again, it is all because of complementary. So for up here, a C is the only thing that combined to that is a G. So I'm making that side. The only thing that combined to a G is a C. So I make that side. So the complement enables us to keep the sequence of all the nucleotides and the bases consistent. We're mainly talking about prokaryotes. That's why we're using the circular chromosome. There's about 30 different enzymes. We're going to end up talking about 6 of these, I think, in here. And what happens is that we have to have an origin of replication. Now, this is important. Now, we come back to those A's and T's we were talking about, how they have just 2 bonds, hydrogen bonds. So this area here, this origin of replication in the middle, this area is going to have a lot of A's and T's. And those 2 bonds are a lot easier to break than the 3 bonds of the C's and G's. So this is going to be an AT rich region, and the enzyme helicase, so ASE, it means it's an enzyme, will come in and unzip it. There's going to be one going this direction and one going over here and one going over here to unzip the DNA. And since this is the origin of replication, replication is going to start at these points on each strand. Now, before replication can happen, it's got to unzip. So we did the helicase. That was one of our enzymes. Another enzyme is what we call an RNA primer, which is a primase. ACE is an enzyme, and that's what this green piece is. A piece of RNA will go in and bind to the DNA and let it know that, let this enzyme, the DNA polymerase 3, know that this is where it starts replicating. And DNA replicates, it builds the new strand in a 5-3. So I'm going to change the color of my lettering of my pen here, and this will be a 5, and right over here where this arrow is a 3, because it's building 5-3 prime. So right over here, 5, 3, and it builds it in that direction. They build towards the replication fork is what we call these things. So that's what, and these are called the leading strands right here. These are continuous. You'll see that on the next slide. So again, on the leading strand right there, it makes a long, it's a continuous piece that just keeps going and going right along. There's that helicase, it's unzipping it. Here's that RNA polymerase or DNA polymerase 3 that's building the new strands. Now on the lagging strand, since remember, it's got to build 3 to 5. And when we're unzipping it, this is the, let me back up a moment. It'll be a little clearer on the, I back up. On this portion, as it unzips, it has to build in small pieces going this way because it unzips and it only exposes a little bit. And it can, remember, it can only build in 5-3. So it'll build a little bit, then a little bit more unzips. So it'll build in 5-3. And that's what they're talking about over on this slide with the lagging strand. Again, it's got to build at 5-3, and it's in these short segments. Those short segments are called Okazaki fragments. But that's how, so you have a small piece here. You'll have a small piece here, small piece here. You'll end up with a small piece over here, and so forth. Now we're back into this. I'm going to change the color again, just because the blue is not showing up that great. At the with all these little pieces of these on these lagging strands, you'll have a DNA polymerase 1 that will come in and remove. Now, every time it has to start, just like here with these green primers, these dark magenta, these magenta squares are also the primers. So DNA polymerase 1 will come in and remove those primers and connect the two strands together. Remove that primer, connect the next strand to it, and so forth. Then at the end, whenever this unzips all the way this direction, and this one unzips all the way that direction, another enzyme called ligase will come in and nick everything together and put it all so you have complete segments, and then you'll have two new copies of DNA. And that's what they're showing you in this picture. They get nicked together and you end up with two new copies. So again, we've gone through, there's 1, 2, actually, that's not one. 1, 2, 3, 4, 5 of the 30 enzymes so far. That goes into replicating DNA. Again, the helicase going there, DNA polymerase 3, building 5 to 3, but you've got to have your primer come in there. Polymerase 1 will remove these primers and hook the fragments together, and well, remove the primers and get them, replace those primers with the DNA sequences instead of RNA sequences. And then the ligase will come in and connect all of those little pieces together. So that you end up with 2 new strands of DNA. The one that we didn't mention was right here, this gyrase, and that's going to help it coil up. A couple of things I want you to note here, though, is that polymerase 3 will proofread, and polymerase 1 also repair some mismatchings also. Ligase can do some repairing also. So there, we have some mechanisms in here, just in case things don't go together correctly. We have a few mechanisms in here to fix any mistakes, to minimize the number of mutations that we do get, because mutations do happen. All right. And because this is when DNA is being copied, it's copying about 750 base pairs per second. So there's a lot going on in it. And depending upon the organism, prokaryotes tend to have a few more mistakes than eukaryotes do, or their proofreading is not as accurate as the eukaryotes, but you can have mistakes, every 10 million base pairs to every billion base pairs. It doesn't sound like very much, but considering all the cells that are in your body and they're copied all the time, they can add up. And that's just on a eukaryote. All right, so on our next concept check, why must the lagging strand of DNA be replicated in short pieces? And well, it's because it can only be copied in one direction. It reads it from 3 to 5, but it builds it 5 to 3. Now, what are we going to do with all this information that we have? All right, so we need to take the information in DNA, which is what we're talking about right here, and turn it into a usable form, because DNA is just storing the information. And when we copy that and turn it into RNA, so DNA to RNA, we call that transcription. And transcription has to happen before translation, because C, an easy way to remember that is that C is before L. So transcription has to happen first, and that is copying the DNA and turning it into all of our RNA molecules that we need. Primarily, we're talking about mRNA, but we still have to create all of our RNA molecules. Then translation is taking this RNA molecule and turning it into proteins, which are the actual things that function in cells. And so that's this next portion right here, the translation, taking the mRNA with the help of tRNA and ribosomal RNA and making a protein. And that's the functional part of life. All right, so how do we do this? Well, DNA is double-stranded. So we're showing you here with one right here and one right here, 2 strands. And we do this in groups of 3s that we call triplets or codons on DNA. Triplets on DNA, but it could be called a codon. And on mRNA, it's called a codon. And on mRNA, it's only going to copy one of the strands, not both of them, because it's a single-stranded molecule. Well, these codons are going to hold the information to find an amino acid, right? And so this one has an amino acid, and since protein is a polymer, a many molecule product, and it's made of all these amino acids, these bonds right here between them are called peptide bonds, and they put peptide bonds in there between the amino acid. You get what we call a polypeptide or a protein. So again, this is the DNA has the original code. We transcribe it into mRNA, And then we will translate that into the sequence of amino acids, which is going to make a protein. And DNA, again, is the blueprint for this. All right. The DNA itself doesn't do anything. It's just holding the information for us. All right. So on these RNAs, we have 4 types. Most of you have probably learned about 3 of them, the messenger, the transfer, and ribosomal. But remember, for replicating, we need primers, and those are, again, priming the DNA. So RNA is a single-stranded molecule in on living organisms, and it has a 5-carbon sugar called ribose. Now, it's not a deoxyribose, which means it has an extra oxygen molecule in there, no biggie. It's got a phosphate in there, just like on RNA. So again, phosphate that's going to bind to the 5' and the 3', and then over here's that 1', and that's where the base is going to come off. And the base that is unique to RNA is uracil. So I'll put the U in there. This is ribose. This is the sugar ribose, and this is the phosphate. It looks just like the DNA nucleotide, except that it's using ribose instead of deoxyribose, and it could have the nitrogenous base of uracil instead of thymine. Messenger RNA, probably the one that most people know about. This one here is carrying the information from DNA. All right, and it's in those codes, those triplet codes called the codon. So there's 1 codon, 2, there's the 3 base pairs for it, 3 base pairs for that one, and so forth. Again, it copies DNA, so it transcribes it and makes a single-stranded copy. This will then another type is tRNA. tRNA is a short one, a shorter strand. It has amino acids will bind to one end of it. We call them a clover leaf because they have these 3 petals. But this is the main thing that we're looking for. It has the anticodon, so the opposite of the codon. So let's say we have UAC on mRNA, that would be AUG. So this is the anticodon to that codon, and it would bind to it, and it brings the amino acid with it so that we can build to the ribosomes, because that's where we're going to build proteins. And so that's the purpose of tRNA. Ribosomes, for ribosomal RNA, makes up the majority of a ribosome is ribosomal RNA. All right. Now there's some protein also involved in making this molecule. And in prokaryotes, there's this whole thing with the large and small unit is a 70S, and eukaryotes an 80S. So there's a little bit difference in the size of them. But the ribosome is where proteins are made, and that's the important part on it. Again, just like on DNA, it will read from 5' to 3'. All right. So first of all, when we're making this stuff, remember, we're talking about transcription, the very beginning of it. So transcription is C comes first. So instead of DNA polymerase, we're using RNA because we're building RNA. It finds a promoter region, tell them it's going to do it, and it's going to copy, again, in that 5 to 3 direction and build it that way. Instead of thymine, we'll put in uracil for it. It has a start codon or initiator codon. It also has terminator codons that tells it when to stop. And depending upon the protein, they can be, 1,000 to 1,200 bases long. Again, depending upon the protein we're talking about. This is the process of transcription. You see you have there's a promoter region here. The RNA polymerase comes in and reads just one side of the DNA, what we call the template strand. So in this case, they're saying the template strand is this blue one. So they're reading it from that direction. But again, they're building 5 to 3, which is very important. You have all these nucleotides sitting there to go in and put it in there, and it builds it. There'll be an initiator codon over here wherever it started, and that's where it'll start building it. And then at the end, there will be a terminator codon, and which will release the mRNA to go to the ribosomes to build a protein. All right, so translation is what we're talking about now. The L comes after C, so we have to transcribe before we can translate. And then again, this is where the mRNA and anything else we need to build it is at the goes to the ribosome. So again, there's the mRNA. We're going to copy it now, or excuse me, it's been transcribed. Now we're going to translate it into a protein. There's tRNA involved, so that we have that, and then don't forget there's rRNA involved with the ribosome itself. So we have the initiation, in other words, the start of building it. The elongation is the actual building of the proteins. We have the end of the protein where it's released, and then we got to fold it and process it. So that way it needs to be folded so that it can form a specific function. If they're misfolded, they don't work right, and we call that mutations because they can be detrimental, most likely. And the master code, this is universal among all organisms, all right? So UUU is codes for phenylalanine in all living things. If you were to count all these, there are 20 amino acids. Right. And you can go from DNA to RNA to a protein, but you can't go from a protein backwards. And the reason why is, let's say the last enzyme or amino acid on a protein was proline. Okay, well, which one was it? Was it CCU, CCC, CCA, CCG? So that's what they're talking about, this redundancy. There's multiple things. Now, some of them only have one or two, like here, your start codon, there's only one. Most of them have at least two, like lysine here, you got 2 there, serine, there's 2 here, but there's also 4 more over here for serine. So there's a lot of redundancy in this, so that even if we had a few mistakes, we can still build that protein, because there's lots of different ways we can make we can code for a serine molecule or an arginine amino acid. So that's good in that aspect. So even with some minor mistakes, it'll still make the protein work, which is very important. Again, transcription is going from DNA to RNA. That's so this arrow right here that we're talking about, that is transcription. All right. Then we go to translation, which is going from the RNA to the protein. We have to use the tRNAs, and then there's going to bring, because they're bringing in the amino acids, and that's what's going to build our polypeptide here, which is a protein. Translation occurs, again, starting with the 5' end, because we always read 5 to 3. We'll get our start codon. Our start codon, again, the mRNA will slide into the ribosome here. It starts at the A site, and it'll slide over to the P site. And at this P site here is where on the A site, the amine site is where the amino acid comes in and bonds, and it'll slide over with it because it's connected with the tRNA. When it gets to the P site, we call that a peptide site, because it's going to form a peptide bond between this amino acid and this amino acid. So we'll end up with the bond between those. And then E is the exit site, so it'll slide out and leave, and we can move a new tRNA in there. And that's what they're showing you on this slide. The first one is that formal methionine for prokaryotes. Slides over, we're going to build a bond between the first 2 amino acids. So there's that bond. Once that bond is made, that the first amino acid is released from the tRNA, and then those 2 amino acids are held together, and this first tRNA is going to go to the exit and then leave. So they're showing it. It's going to go back out to the cytoplasm and pick up another amino acid and be ready to come back down again. So now that it's gone, this tRNA can slide over to the P site. All right. And another amino acid, another tRNA will come in to the A site, to the amine site. You form another peptide bond, which basically is going to allow the second tRNA to be released. And then the third one will slide over. A fourth one will come in. You're going to form another. So we're starting to get all of these amino acids bound together. And we're building it. And that's going to be, so we have many peptide bonds, polypeptide, which is protein, until we get down there to that stop codon, which I believe is on the next slide. I didn't specify, but when we get to the stop codon does not code for any amino acids. So whenever it slides over to the P site, there's nothing for it to bond to, or when the last one, when this one moves into the A site and the second to last one moves over here, there's nothing for it to bond to. So it releases the protein out, and then the protein will get folded and processed to go do whatever it needs to do. And that's how proteins are formed. That's protein synthesis and the translation portion of it anyway. Yeah, we have 3 termination codons. I'm not going to have you memorize those because there's no tRNA that goes to it because there's no amino acid that goes to those. So then it releases the polypeptide, which is the protein. Now, a polyribosomal complex is poly means many. So we have many ribosomes going on this. And how this works is that the mRNA, the yellow strand right here, enters the first ribosome, starts building it. And as it's passing through, another ribosome can come in and then it'll pass through it and starts building another protein. So for every ribosome you see here, it's going to build another protein because you need more than just one of those things. And this way they can build lots of proteins at one time off of one strand of mRNA. So you don't have to make multiple strands to make multiple proteins. You can make multiple ones out of one. It's just more efficient that way. Now in eukaryotic cells, you cannot do transcription and translation at the same time. And the reason why is because transcription with the copy of the DNA occurs in the nucleus while translation is in the cytoplasm. Now in prokaryotes, they don't have a nucleus. So the cytoplasm is right there around the DNA. So as it's making the mRNA, it can actually start translating it at the same time. Eukaryotes can't do that. Eukaryote has the AUG start codon just like prokaryotes, but prokaryotes use formal methionine and eukaryotes just use methionine. In eukaryotes, the mRNA is for a single protein, and in bacteria, you can get many, a lot, we're going to talk about some a little bit later that are going to code for, one strand of mRNA is going to code for like 3 proteins. Eukaryotic DNA also has a portion of what we call introns, and these are non-coding sequences. When they were first discovered, they called them junk DNA, but we found out that they do actually do a little bit, but they're just not coding for any proteins. And these non-coding portions must be spliced out before mRNA can make proteins in eukaryotes. So it is different making a protein in a prokaryote than it is a eukaryote. So the removing of those introns we were talking about. So here they are. The introns are the green portions. And intron, think of it as the insert, they're inserted in. And exons, think of these as being the expressed. So these are the ones that are going to get made into, dang it, terrible S there. These are the ones that are going to be made into the protein. And this is all occurring in the nucleus. It's very important there. There's a protein called a spliceosome that will go through and remove those introns, and it'll put the exons together. And this is the mRNA that gets copied into, or gets translated into a protein. It will leave the nucleus and go out to the cytoplasm where it can go to the ribosomes and be translated. But the introns had to be removed first. So remember, exons are the expressed portions of the DNA, and introns, these are just insertions into the DNA, and there's spaces in there. Again, originally they were called junk DNA, but we don't call them that anymore. They're introns and exons. All right. Now, we don't just copy genes all the time. We don't just make proteins all the time. They need to be regulated. And portions that regulate this are called operons. Okay, now we have a couple different types. We have an inducible type. These are ones that, again, get turned on when we have a bunch of substrate. And the repressible ones that get turned off when we have a lot of product. And it kind of makes sense with this a little bit. So let's jump into some of these. So our first one, well, to do these operators, you have to have a regulator, you have to have your controller, and then the actual structural genes. The regulator is just the portion of it that is going to be either turning things on or turning things on, or off, excuse me. We're going to start by the ones that turn them on. And then you have a promoter portion. So this is where the polymerase is actually looking to make the copies of it. And then the structural portion of it are the actual genes that get turned into proteins. So those are the three parts that you need for operons to be working. The first one, and this is probably the most studied operon. It's been studied for 60 years or so now. This is the lac operon for lactose. So when there's no lactose present, you don't need to be making these proteins. So you can save energy that way. All right, so in this case, the regulator gene is being active because it is stopping RNA polymerase from copying the genes. All right, so this one is a regulator one, and it is it's keeping you from producing the making the proteins. What happens is that I guess it's an inducer. Well, no, to me, lactose is the inducer. When you have lactose, and that's what they're showing you here, lactase will go in, and that regulator, that repressor gene, it will change its shape so that it releases and lets the RNA polymerase go ahead and copy everything. So as long as you have lactose present, it will allow these genes to be, these proteins to be made. When the lactose levels get below a certain amount, the repressor will go over and block it, and you won't make those genes anymore. So you've got to have lactose present. So that's why this needs to induce. You put it there, and it'll induce this to happen. All right, so yeah, that's what they're showing you. Now, okay, now we're going with the arginine, the operons. These are repressible ones. And in this case, the proteins are being made all the time. Now, whenever you get plenty of arginine right here, plenty of it produced, then it will go and activate the repressor, and the repressor will go and block it, so that, because you have plenty of it, you don't need any more. So it'll block it and keep you from making these proteins. So it works just the opposite of the lac operon, the arginine operon does. So right here, see, the arginine is going binding to the repressor. So it acts as a co-repressor, because you need both of them. And it blocks the RNA polymerase from copying those genes. So it stopped the protein synthesis because you have plenty of the product. You don't need any more. When that product gets used up, then it'll release it, and you'll start making more. You'll start making more of those proteins to make more of those that product. All right, so our concept on this one If an operant's repressor is active, so it's repressing you, it's not letting you make proteins, what's happening? That means that transcription is not occurring. So if it's active, so we can back up. This we're talking about right here. This is an active repressor, so it's not letting you make proteins. And on this, when it becomes inactive, you can make the proteins. So over here, it's inactive, so you're making proteins. When it becomes active, it stops you from making proteins. So if it's active, you're not making any proteins, not transcribing. Mutations now, changes in the DNA. Any change that gets passed on is called the mutation. All right. What we expect to see in the wild, we call these wild types, the wild strains. And if they have a different trait, we call that a mutant strain. And that could be just the color of the cell. It could have a different structure, like a flagella or something on it. So that's the characteristics. It could process a different type of sugar. It could become antibiotic resistant. All of those would be mutant strains. There's all different kinds of things that could be that. And that's the difference. The wild type is what we expect. And anything that's out of the ordinary would be called a mutant strain. Most of the time we could pick these up from using a loop, but they do have what we call replica plating here, where you keep originals. So you grow a species going out here and you see something different here. So you'll get it's a press that you'll push down on it so that each one of these will grow in the exact same spot. Then you use two different plates. One of them is like missing a nutrient. So it's going to favor one strain over a different strain, is what we're looking for. So as you see, when you grow it, the mutant strain is not there. It is gone. So now we can pick up that mutant strain and grow a complete mutant colony out of it and have nothing but the mutant in there. And then we can run tests on the mutants. All right. Now, what types of mutations do we have? Ones that occur randomly, and these again, remember there's going to be a mistake every so often when you're replicating. These are called spontaneous mutations. We don't know what's causing them, they just happen. Ones that we cause to happen, we call those the inducible ones. And we know certain things that can cause them, and those are called mutagens. And here's a few things that will cause mutations in bacteria. So we know they're going to cause most likely going to cause mutations in us too if we were around it too much. And it's like here, ultraviolet's a big one. What does ultraviolet do to us if we're exposed to it over long periods of time? It causes cancer. It's mutations, and here's what happens. It's what it does to it. So you want small amounts of UV light because it'll cause problems with even in bacteria. And if it's exposed enough, it can kill them there. We know that gamma rays kill and X-rays kill, and if you have enough exposure to them. So it does vary, the amount of exposure anyway. All right, so on these, we have most of these are going to be point mutations, okay? In other words, we're talking about a single base. We can add a base, we can delete a base, we can, again, addition, deletion, or we can even substitute, all right? We can add a new one in there, we can remove one, or we can change one. All right, so missense mutations, these are ones that are going to cause, these are changing a amino acid or changing a base, which is going to cause an amino acid to change. But we're just talking about one, so that's a missense. Your nonsense causes a normal codon that's coding for an amino acid to turn into a stop codon, so there is no amino acid for it. Silent mutations are ones that, yes, it does change the base, But because of that redundancy we talked about earlier, the amino acid doesn't change, so we don't notice any effect on these. So that's why they're silent. Back mutations are ones that, because there are some mutations that we know about that occur, and they're real easy to flip back. So that's what we call a back mutation. A frame shift is where we're going to be doing, on a frame shift, that is the addition or deletion, because you're moving the lines around on it. So this is what that's a frame shift mutation. We got some examples coming up here in a moment. And that's what we're talking about here. So in the wild, the original, what we expect if we were to read it, and that's what they're giving you in this 3 bases, so those 3 codons, or the codon. The big bad dog ate the fat red cat. So in a substitution, if we have a missense where it's just changing one, but doesn't do anything else, it changes what the amino acid is, they're showing you right here, it changed that one base, that I, from fat to fit. So now it changed the protein, the product that came out of it with that one change. But the length stayed the same. A nonsense is where it went and it changed what would have been dog to a stop codon, so it stops. So now you've changed the length of your protein, and it's not going to be effective. This is a negative. This is most likely going to be a negative change. A frame shift. So again, we have big bad dog. So they inserted a B there. Now look, it doesn't make sense, the rest of it, because you shifted the frame over, and that's from an insertion by adding one. And we can also remove one. We pulled the A out, so it shifted there. It doesn't make sense. These are usually non-functional proteins, and non-functional proteins are almost always, I shouldn't say almost always fatal, but can be fatal. All right, so up here, this would be a non-functioning protein here also. This one is just going to be a different protein. It might not function correctly, but it could still be functioning on this particular one. So these are the types of mutations: the missense, where it doesn't quite make sense; the nonsense, where it stops; and then your insertion and deletion, which are both frame shift. Remember, we talked about some things that can repair mutations. We talked about the DNA polymerase that proofreads these. There's another enzyme in there that does mismatch pairs that'll, because sometimes you'll get an A that bonds to a C instead of a T like it should. So you could go through and fix that. UV damage, if it's not too much, there's a light repair enzyme that can go in and fix that. As long as it's not too much. And then you have some excision types that will go in and actually remove segments so that the nucleotides line up in the same space. So these are the excision ones, are more dealing with the frame shift ones, removing portions that aren't making sense. Now, testing this stuff, testing for mutagens, we use the Ames test to do this. This is a very common one in the industry to do this. And what they do is they use a salmonella strain that we know is it's easily mutated. And this particular one has lost the ability to make histidine, which is an amino acid. But because it's a common one, it's actually able to do that back mutation where it goes back to the original. So, and it does it at a certain rate. So we expect to see it happen. So they're showing you up here, you inoculate the medium with the bacteria. We expect to see some mutation where it can actually use the histidine. If we see a bunch of it, so it's really increased the amount of back mutation, then that means whatever that chemical is most likely a mutagen, which can cause mutation. Now, again, this is one that will happen. This is a back mutation that is really susceptible. It happens real often, and we're speeding that process up, which just means we're speeding up mutations. So that's one way they test for mutations. All right, mutations, if they're negative, if they lead to non-functions, most likely they are harmful or fatal. And if it's beneficial, though, then it's going to help it to survive. And these are the ones that are going to change populations, which lead to evolution. So again, it's going to be selected for, which is natural selection. And again, that is how evolution works. It's small changes acquired, multiple changes picked up little at a time over a long period of time to get us to mutate or to get new species. So of the mutations, which of these mutations would cause a frame shift mutation? So there's the frame shifts are where it's going to move it from one sense to another sense. And out of these is the deletion mutation. All right, so recombination of events. What's a quick way for prokaryotes to change their DNA? There's three methods for it. Now we have them right here: conjugation, transformation, and transduction. And what they're doing is they're getting DNA from another organism. It's not sexual reproduction because prokaryotes do not do sexual reproduction, but this is ways that they can get DNA from other organisms. The first one is conjugation. Conjugation, in simple terms, one gene has the ability to share, one prokaryote has the ability to send a gene or a fragment of DNA, most likely a plasmid, across to another prokaryote, to another organism. Could be the same species, could be a similar genus, could be completely different. All right. Most likely they're pretty similar to each other. And by doing it, and we call this the F plus, and what happens is that's going to end up with an F plus also. So again, it transfers that information from it's only one direction. It is not both directions. And this is conjugation. Okay. There's a couple of methods of it. High frequency on them. It's getting paired up into the bacterial genome and not just the plasmid sitting out there. So there is a couple of methods of it, but really, ultimately, what I want you to know with conjugation is that, again, it is the genes are going from one to another. So again, this one is going this direction. Now this one has that same trait that this one had. Okay, but again, it is only going from one to the other, not back and forth. So conjugation is just one direction on that. But it is a way for the new species here, or the other organism, to gain traits without reproducing. Another one is transformation. Transformation is where you have a lyse cell. So in other words, another cell that is broken apart. You get fragments of its DNA out in the environment. And a living cell can acquire this genome, and it can incorporate. It could be a plasmid out here in it, or it could put it into the DNA. So again, it gathered new DNA. Again, not related to, or could be related, unrelated. But again, it acquired new DNA, so this has new ability, and it's a way for them, again, to change for mutate. It's again, get new DNA, excuse me, so it can survive better. Maybe it won't survive better, but if it does survive better, then they're going to reproduce more and so forth. Again, it's for genetic variability in prokaryotes. This is most famously done with Griffin, I believe it was in the 1920s when this occurred. And what he did was he took a wild type cell that had a capsule on it, and they called it the S strain because it was smooth when you looked at it. And it was Streptococcus pneumoniae, I believe the bacteria was. Injected it into a mouse, the mouse got sick and died. He took another strain without a capsule, so it looked rough when you looked at the colonies growing on the plate. They weren't nice and smooth, they were rough. So hence R strain. Again, same species, Streptococcus pneumoniae, injected it into the mouse. The mouse survived. So another one, then he killed the smooth one, the S strain, injected it into the mouse. The mouse survived. Another one, he killed the S strain, and he added the live R strain, the rough ones, put both of them into the mouse died. And when he cultured the bacteria, some of them came out smooth, which proved that they picked up the ability to grow this capsule around them to make them look smooth from the environment, because these were dead cells. So it's not like they were passing them on to conjugation or something. So the rough ones, some of the rough ones picked up these that ability from the environment, and which proved transformation. And transduction, we've mentioned before. We were doing viruses, and this is where you get fragments of the host DNA is picked up in the phage, the virus, and then it's carried on whenever the virus lyses the bacteria and goes and infects a new bacteria. Part of that genome can be carried with it and incorporated into the new bacteria, newly infected bacteria, so that bacteria picks up that trait. So it gains DNA through a bacterial vector, or excuse me, a viral vector from a different bacteria. Usually they're very similar to each other, because viruses are fairly specific on what organisms they can infect, so they're going to be related to each other, most likely on transduction. And they're showing you that in this slide here. So it affects it when they're all forming pieces of right there. They're showing it right there. Pieces of the host genome gets incorporated into the capsule or the capsid, and then gets inserted into the new one. All right, so I get right there. And again, they're showing you over here. There's a couple of different ways of it happening, but basically it is picking it up from a virus and then passed on to another bacteria. All right, so again, in your chapter 9, your textbook, there's a table 9.5 that talks about these different types. Again, cells lyse, picks it up. These are indirect because they don't have direct contact with that, but with the donating organism, unlike the conjugation. Another way for things for mutations and genetic variability is transposons, and these are what we call jumping genes. And what it is, it's a segment of a genome that will move, and sometimes it just plain moves. Sometimes it'll copy itself and then move, so you end up with 2 strands of it. Sometimes, like on the bacteria, it can leave the main DNA and go to the plasmid. Plasmids can leave. This, again, depending upon what the gene and how it's doing it and where it does it, it can be beneficial or it can be harmful. If it's beneficial, again, it's going to be passed on. If it's harmful, most likely it's going to die and not be passed on. So again, this is just genes moving around, so you get different combinations of it, because it can be, because you're going to change the sequences a little bit. And again, it can go from one side of the chromosome to another. It can go from a chromosome to a plasmid, go from plasmid to a chromosome, all these different methods. And those are jumping genes. All right, so coming back to our concept here. If an F+ cell, so this means that F+ means it can form the pilus to do conjugation. So which one? Yeah, it can undergo conjugation, B, because F+, it's able to form that pilus. All right, that's it. Have a good day.